inaugural HERN webinar series. HERN is the Human Eyelid Research Network, and my name is John Cadiz. Today we're going to be introducing you to HERN briefly, because we know that many of you may not know what the Human Eyelid Research Network is, and some of you may have heard of it. And then we're going to introduce you to our presenters. Next slide. Next slide, please. So HERN is composed of 6 different groups. Each has a different theme that they research. And what the NIH did back in 2014 was it brought some of the world's leading scientists in diabetes together and organized them by various themes. And you can see those themes here. One of them is called CHIB. It's largely an engineering group. They try to put either tissue on a chip and try to understand how a beta cell might work and assay it. You have a group called CTAR, the Consortium of Targeting and Regeneration, understanding how you could target certain cells in your body. And we're going to be hearing from a project there today. You have another consortium called the Consortium on Beta Cell Death and Survival, and that's really just looking at that beta cell, how to keep that alive, how to grow more cells. There's a program called the Human Pancreas Analysis Consortium, and that's a group of people that heavily characterize the pancreas using a variety of different assays and methodologies, and then makes those data sets available to researchers to study. And then there is the Consortium on Modeling of Autoimmune Interaction, and largely that is trying to understand the immunology of diabetes. And then I am part of a group called the HIREC, which is the Human Eyelid Research Enhancement Center, and that largely tries to organize our group, and that's ran by myself, Joyce Nyland, and Layla Rouse. Next slide. So I won't cover what all the different science that's happening there. We have some reports that we write. They're in non-scientific terms. You can browse through each of these different groups and what they're contributing. Next slide. And there's also a lot of data that this group generates as well too. And there are different databases where a lot of this information is stored. Next slide. And these databases you can find on through the HERN website, which is hernnetwork.org. But there you'll find some of the different resources that you'll hear about today and many others. So I'll encourage you to go there. But for today, what we're going to be talking about is we're going to be talking about a really interesting project. Next slide, please. And that has to do with the different resources that HERN generates. So, you know, whether we're talking about technologies or bioreagents or transcriptomics data sets, there's a lot of different opportunities both to interact with HERN and also use some of these tools for your own research. So today we have 2 very distinguished speakers who are going to talk to us about adeno-associated viral vectors. The first is Dr. Mark Kaye. He's the professor of pediatrics and genetics at Stanford University. He's the division head of human gene therapy. He was one of the founders of the American Society of Gene Therapy. And he actually started studying, Mark, as I understand it, started studying Drosophila genetics, where he first got interested in genetics and soon led to his passion in developmental genetics. And next is the next speaker that we'll hear after him is Dr. Nakai, who is at the Oregon Health Sciences University. He's a professor of molecular and medical genetics. He is a professor of molecular microbiology and immunology, and a distinguished professor in molecular medicine. And he's been interested in AAV vectors from the 1990s, where I guess he first started looking at hemophilia. So these two guys are well-versed, know this area very well. So without further ado, I'd like to turn it over to you, Mark. Thank you very much for agreeing to speak with us today.
So here's a conflict of interest disclosure slide for both myself and Nakai. I'm fortunate that a lot of my knowledge in AAV started when Nakai was actually a postdoc in my lab many years ago, and now he's gone on and done tremendous work at Oregon Health Sciences. What we plan to try to cover here, and I'll spend the first 30 minutes or so going through a number of topics, and then Nakai will present some of the latest cutting edge technologies and also discuss the resources available to investigators. But what I wanted to do is cover the vectors in clinical medicine, some applications of the vector, and ways that we try to understand vector biology. And of course, we can't cover everything in this short period of time. And I'll talk about things in general. I'll talk about some of the work that we've done as well. Now, for gene therapy, one of the most important aspects is the vector. That is what delivers the sequences into the desired cells for therapeutic intervention. And there's a number of different vector systems. They all have their strengths and weaknesses, but we will focus on the AAV or adeno-associated viruses that are shown here. Over the years, if you look at the number of vectors that have been used, or the percentage of vectors that are used in the current clinical trials, you can see that AAV represents just under 8% or so. But that percentage is growing over the years as AAV has become more popular, especially for uses for direct in vivo delivery into humans. And if you look at the examples of some of the AAV trials, and last I looked recently, there were over 200 trials that have either been completed or are starting. They cover a wide range of disorders. Retinal dystrophy was really the first licensed product in the United States. There was a previous approved product, for lipoprotein lipase deficiency in Europe. It wasn't approved in the U.S., and subsequently it's no longer being produced. I think there was some questions about efficacy, and it's an extremely rare disorder. Spinal muscular atrophy, SMA, there's a AAV vector for that. That's also been FDA approved. And there's been a lot of remarkable outcomes with this. But other disorders that are in various stages of testing include things like the hemophilias, Parkinson's disease, muscular dystrophies, including DMD, Pompe's disease, Alzheimer's, a number of inborn metabolic diseases such as Crigler-Najjar, glycogen storage, PKU, Sanfilippo, Fabry's disease, and there have been some gene therapy trials using AAV for heart failure, and there's some more coming down the line soon. Now, a little bit about AAV biology. The wild-type virus itself contains two genes, rep and cap, really two proteins or classes of proteins. Rep is involved in replicating the viral genomes, and cap is involved in encapsulating the viral genome. So what we do as gene therapists are we supply rep and cap and some of the other functional genes required for replication that come from other helper virus sequences. And we basically package the therapeutic gene, usually with a control element like a promoter and polyadenylation signal. Or now, and well, I'll show some examples where you may not even be expressing a gene, but want to deliver a piece of DNA that's involved in some aspect of genome editing. And the important point is that in the vector itself, all the viral proteins are removed, the viral encoding genes are removed, and there's only the ITRs that remain, which are short structural sequences. Now, when AAV transduces a cell, that each AAV particle consists of a single-stranded genome sequence, so it's single-stranded DNA. And when you make AAV vectors, they have an equal amount of the plus and the minus strand. And these get into the cell and they get into the nucleus where they become, they become double-stranded, and then they can take on different forms. The majority of them, again, are episomal, and this is important, as you'll see later, because the AAVs, by and large, don't integrate. Some of them become monomeric circles, and some become concatemers. And again, these are responsible for the transgene expression. It's important to note that a very small proportion of the AAV genomes will integrate. And the exact number isn't clear, but it's probably well under 1%, something like 0.1% or even lower than that. And although the percentage is low, you'll see later that this can be something that people need to consider and deal with. Now, many years ago, for example, we actually published some papers back in the mid-90s showing that if you took an AAV vector that expressed human factor IX, you could inject it into mice and get a dose response and get persistent therapeutic curative levels of factor IX, again, that persisted for the life of the mouse. And this was very exciting because up until this point, we either had vectors that gave subtherapeutic transgene product, or we could get transgene products but with severe toxicity that would not make them clinically amenable. And then what people learned was that there were different variants in the capsids that were really affected the percentage of cells that could get transduced. And when you compared AAV2 with AAV8, even in the early days, there was substantial difference in transduction, even with the same dose carrying the same transgene. So this showed that small sequence differences, because AAV2 and AAV8 are 82% conserved at the amino acid sequence, there was a lot of variation in transduction. And I use hemophilia as one example because this is one of the diseases for which there's a long history. And you can start looking back in, again, the early 1990s with the original production of recombinant AAV vectors. The first in human dosing included a skeletal muscle delivery where the efficiency and the levels of persistent factor IX weren't sufficient. And then things started to happen in humans. We actually did a study it was a collaboration between myself, Kathy High, and Avogen, a company where we, for the first time, injected AAV systemically into humans, and lots of things were learned in humans that weren't anticipated based on all the other animal models. But since then now, things have improved. There have been a number of trials that look very promising for AAV. AAV human factor IX, the most advanced trials are Spark Therapeutics, and there's been, these studies have been published. Unicure and some other companies are also have variants on the AAV factor 9 vector. And also AAV factor 8, BioMarin has the longest data, Spark, Sangamo, and now there's some other trials as well. And you can see that with in a relatively safe manner for factor 9, you can get long-term expression that would be therapeutic and really ameliorate the bleeding diathesis in these patients. One of the things that has come out of AAV research is the fact that AAV has very different transduction in different animals. And what you see here is that it's been very difficult to actually establish how to go from an animal to a human. And there have been lots of different ideas about this, and really we don't know what the best animal models were. I sometimes think about if we were around during Noah's Ark's time, meaning the AAV biologists were around at that point, we would have the perfect way to study this, as we would have had at least N of 2 for every species that we could do the tests on. But clearly, this has been an issue of major study over the years. And one of the things we were interested in was using a mouse model that was developed in Marcus Grumpy's lab, and where you can take a mouse that has a genetic defect and you can actually implant into the liver human hepatocytes, primary human hepatocytes that repopulate the mouse. And you can actually then study AAV vectors in terms of their ability to transduce. And what we found was that based on the human clinical data that had been published with AAV2 and 8, I mean, studies that we participated in, the humanized mouse, at least in these two examples, appeared to recapitulate what we saw in the human, which the mouse really didn't. And then we utilized these animal models, and what we did was we made complex AAV libraries in which we had we made libraries that, and I'll talk a little bit about this in a minute, in which we took various different capsids, the DNA sequences, we chopped them up into small pieces, religate them, and then we would make a library, let's say, of 1 million variants, each one having its own capsid. And then we passaged this through the humanized mouse system because we wanted something that would work best in a human liver. And what we show here is that when we were able to do this selection, we found that a variant called LKO3 that's made up of about 4 different capsids, chimeric. Again, this was all made through selection through this humanized mouse system. And when we tested for its ability to transduce the human hepatocytes in a mouse model, so this is the humanized mouse, we found that LKO3 transduced at about 10 to 15 times better than AAV8. And I'll point out that one of the interesting findings was that LKO3 itself, and then subsequently some other capsids that we've isolated in subsequent screens, don't work well in mice, and they appear to be primate specific. But Katja Purkin in the lab actually made more complex libraries through this DNA shuffling. And she made 2 different libraries, starting with capsids that had either 10 or 18 parents. And this is kind of the way we do things now, is that we take our shuffling library and we do both DNA sequence, we put a DNA barcode in. I mean, this is something that Nakai originally had done, and now, as he'll mention, has even more sophisticated ways of doing this. But we put a DNA linker into the capsid library so that each linker or each barcode is unique to the capsid. And then we also do full-length single molecule DNA sequencing using PacBio platform. And then we can get a lot of information from our screens, and we can get the data much more quickly than we were able to do previously with the earlier screens where we had to do Sanger sequencing. So one of the applications of this was to passage the library through human islets, and we used intact islets and dissociated islets, and we did multiple rounds of selection, and we came up with 3 capsids labeled KP1, 2, and 3. And I won't go through all of the data. It's been recently published, but we found that there was one set of capsids, like the KP1 that I mentioned, that has better transduction in human islets than any of the other previously tested capsids. And this has really resulted in a large improvement in transduction. And one of our goals are to use the AAV in humans and to actually inject it through the pancreatic duct to transduce islet cells where we can do reprogramming, such as in type 1 diabetes, or deliver a protein that can reduce immune responses that can hopefully abate the autoimmunity associated with type 1 diabetes. Now, AAV has its limitations as well. I don't want to make it sound like it's a panacea, but one of the limitations is genome size. The total packaging is just under 5 kb, and you need to have about 300 base pairs or sequences for the ITRs. Manufacturing of some of the various capsids have been difficult. There's many different platforms now for manufacturing, and there's a lot of I guess there's a lot of things that are unclear yet about whether manufacturing, let's say, in baculovirus insect cells versus mammalian cells result in equivalent outcomes. The purification after the production varies from place to place, and again, how this affects transduction and safety is still unclear. As I mentioned, AAV is episomal, so when the cells turn over, you'll lose the expression. There's issues of pre-existing immunity. Many people have been exposed to AAV, and a certain percentage of the population won't be amenable to systemic delivery because transduction will be blocked, and the relative abundance of neutralizing antibodies can vary from capsid to capsid. And even if you are able to be transduced, there's really inability to re-administer after systemic administration. There can be some issues because, as I mentioned, there is some integration, and there have been studies in neonatal mice where the vector can integrate next to a proto-oncogenic sequence. The promoter from the vector then can result in forming tumors, and this is something that has to be watched for. There have now been toxicity in some of the clinical trials, and not all toxicities are equal. There's toxicities that are acute, and again, even within this acute category, there's differences, and what the exact causes are still unclear. There's cell-mediated immune response or something that can result, for example, if you give it to the liver or you get capsids in the liver, that you can get a transaminitis or elevation of liver enzymes that results in the destruction of the transduced cells. Most of the time, this has been abrogated by giving a short course of steroids that are relatively safe. So again, it's not a showstopper, but it is something that has caused some issues. And again, for many of these types of events, Some of these appear to be somewhat human specific, and it's been very difficult to determine the cause and effect. Now, as I mentioned, AAV is episomal, and it's lost with transgene expression. So if you wanted to treat a proliferating tissue, it's a problem, or even a neonate where a lot of the metabolic diseases would be optimally treated. You can show both in mice and now in non-human primates that if you inject early on, you get a high peak of expression that's lost very quickly. So, and again, readministration's a problem. There are lots of clever ways people are trying to use to overcome this. Now, if you can integrate into a progenitor cell or a cell that, as long as it or its daughter cells persist, you could, in theory, get lifelong expression. So there's been a lot of interest in using AAV in genome editing approaches. You can use different nucleases that can actually result in integration at specific loci. And examples could include homing endonucleases, zinc finger nucleases, TALENs, or CRISPR-Cas9. There are, again, plus and minuses of all these systems. And some of the things you may want to do is if you can cut a specific sequence during the repair process, you can get a gene knockout. You can use a template DNA so that where you cut, you can get something called non-homologous end joining. Insert your DNA sequence where the break was, because AAV does tend to integrate into broken regions of DNA. You can make flanking sequences that are homologous to the donor DNA, and again, make a cut to enhance homologous recombination. There are other approaches people are using to do base editing, where you can express a gene that can actually correct a certain base in a mutation. And people have used now dead nucleases where they still bind to the region of DNA, but they have an activation or an inactivating protein, so that you can express the nuclease from an AAV with the appropriate guide RNA, and you can either turn off transcription or enhance transcription. So there's lots of clever ways people are using, and again, these all have therapeutic interventions. Now, as I mentioned, AAV can integrate, and if it integrates in the wrong place, you can get hepatocellular carcinoma. This has been well studied now in neonatal mice. Whether or not this is going to occur in humans is unknown, but it's something to keep in mind. Our lab developed something called, we call it gene ride. So we use AAV for homologous recombination without nucleases. So we design a homology arm to the region we want to integrate the DNA. And in between that, we put a P2A, which is a ribosome skipping site, with the coding transgene product. We can then get homologous recombination at that site. And for example, in the liver, if we target albumin, the end of the albumin gene, such that now when we get a messenger RNA, we get a fused message that makes the albumin itself and the transgene product or therapeutic gene product as shown here. So you can get two proteins from the same transcript. And we use this to treat mice, neonates and adult mice, and we can get therapeutic levels of factor IX and abrogate the bleeding diathesis in hemophilia B mice. Some of the other applications for this, methylmalonic acidemia, which has been studied a lot by Randy Chandler and Chuck Venditti at the NIH. This data will be published soon. There's actually a phase 1/2 clinical trial that will start soon. Crigler-Najjar, again, a lot of this data has come from Andres Muro's lab in Italy, and also alpha-1 antitrypsin deficiency. Now, the efficiency of this is low, so for some diseases, it may still be amenable to get a therapeutic response. But there's lots of ways that we and others are trying to enhance the recombination event. We recently did a unbiased genetic screen and found that if we interfered with the FANC-M complex and some of the proteins that bind, we can get up to a 20-fold enhancement of AAV-HR without nucleases. In primary cells, the enhancement is much lower. It's still there, but we are looking at other drugs and doing other screens so that we can increase. So the idea would be to give a drug for a very short period of time that would enhance the AAV-HR at the time the AAV was given. So we do have improved capsids, and again, using this with GeneRide, we think that we may be able to circumvent some of the limitations of AAV, but again, not all of them. And again, this is work in progress. There's a lot of progress using the nuclease approaches as well that I really don't have time to get into. So with that, I'll stop and I'll hand it over to Nakai.
Hi, Mark. Thank you very much. And I'd like to override your screen. Just give me a minute. So I hope everybody can see my screen. I just changed to slideshow.
Yes, we can see it, Nakai. Thank you.
Hi. So I just would like to take over Mark's talk. And so what I would try to do is just introduce recent advancement in AAV vector technology, but a project supported by NIDDK is aimed at establishing new gene therapy for type 1 diabetes. And the idea is kind of clear, and we have some idea what transcription factors we can introduce to non-beta cells to trans-differentiate non-beta cells to beta cells using AAV. But a complicated thing is how we deliver those kind of transcription factors to the target cells. And it's not as straightforward you might think, and you may think that we can just inject AAV into animals and humans and deliver AAV to the pancreatic islets and trans-differentiate alpha cells, beta cells, but story is not so straightforward. And so that is because the AAV biology really complicated, and we need to think about the many different things that you may not think of. And this is what AAV researchers are trying to figure out. And so one of the complicated thing is this. And so this is just like a picture how AAV can transduce many different tissues. And this gives you idea how AAV travel from the bloodstream to the actual endpoint of the nucleus of the target cells. When you inject AAV vectors, it gets in the bloodstream and it goes out from the bloodstream into the extracellular matrix and then binds AAV receptor and endocytosis and trafficking the cytoplasm and the endosomal escape. And eventually it gets into the nucleus and DNA is released and transcribed and express therapeutic proteins of your interest. But there are a lot of physical barriers. For example, right after AAV is injected bloodstream, it can bind the red blood cells, for example, AAV serotype 4, it can efficiently be sequestered by the blood cells. And the people have been trying to figure out what will happen when we do this kind of things. Then there are several advancement, for example, and the blood concentration, AAV9 stays in the bloodstream really long time, but in contrast, AAV1 is rapidly cleared from the bloodstream. And we used to think that probably pharmacokinetics is the same across different serotypes. But now we know that it's totally different. And we still don't know what the mechanisms behind this one. And also what we know is when you inject AAV into the bloodstream, there are a lot of serum proteins and albumin can interact. And the good thing is, for example, AAV8 can bind albumin and enhance liver transduction. but in the bloodstream, there are several inhibitors. And one of the inhibitor that is well known is galectin-3 binding proteins. And this can bind AAV and inhibit transduction. And for example, in mice, and there is a activator, which is a C-reactive protein in mice, and AAV binds C-reactive protein and enhance transduction, but AAV is not interacting with C-reactive protein in humans. So that means those kind of interactions are really species dependent and also serotype dependent. And also, and the endothelial barrier crossing is really mystery. And one of the example we have, and also other people have recently found is, for example, Ly6A variant, which is only one known protein that can significantly enhance endothelial cell barrier crossing. But AAV, you know, it can cross endothelial cell barrier, but we still don't know how those kind of crossing happens by being mediated by cellular mechanisms, which is still we don't know well. And also, so people have known what receptors AAV2 uses when it binds cell surface, which is a heparan sulfate proteoglycan. And also recently, and not recent, maybe 10 years ago, and people have found that galactose can serve as an AAV serotype 9 primary cell attachment receptor and the stearic acid. And also there are a lot of co-receptors, but still don't know what primary attachment serve as a kind of, how to say, receptor for many different serotypes and mutants. This is still, we don't know well. But recently, there are significant advancement in terms of understanding what cellular proteins are really important for serving as entry factors. And one of the big discovery was made in 2016, maybe 4 years ago, which is a discovery of KIAA0390L as a primary receptor for many different serotypes, which is now called AAV receptor. And also more recently, and people have found that GPR108 is also a really important entry factors important for multiple different serotypes. And so I just would like to focus on this KIAA319L, which is AAVR. And this protein was originally, initially identified at NIH group and led by Kevin Brown and also Jiamin Ku as a serum proteins that can bind AAV serotype 2. But at the point, and people haven't thought that this is really important protein, but Michael Chapman's lab and also John Khaled group and at Stanford, and rediscovered that this protein is really important for as a cellular entry factors for multiple different serotypes. And so this is a screen they did. They identified this AAVR is a really important factor. And when it's knocked out, and you can't get transduction with multiple AAV serotypes. And also this happens many different serotypes, and this is wild type. This is AAVR knockout cell line, and you can really see that if you knock out AAVR, and you can't see transduction. But interestingly, and if you overexpress this AAVR, now you can enhance cell transduction in different serotypes. So that gives us some idea how we can utilize this idea. And in enhancing transduction, I can just talk about a little bit in a second. And then so AAVR is a 110 kilodalton protein that has the ect domain here and a transmembrane domain here. This is basically a membrane protein, and this has the 5 PKD repeat domain. Is a polycystic kidney disease repeat domain. And then, so this serves as a co-receptor of AAV. Primary attachment receptor could be heparan sulfate proteoglycan or galactose, but once AAV binds this primary attachment receptor, AAVR binds also AAV and quickly and internalize inside the cells and traffic it and deliver to the transport system. And they help AAV vectors escape from this transcription and then help AAV vectors to get into the nucleus. But interestingly, so this is universal and sort of vector, but how this protein interact with different cell types is different. And for example, AAV5 uses this PDK1 domain for attachment, and AAV2 use PDK2. PKD2 and AAV1, AAV8 uses combination PKD1 and 2. And interestingly, and Luc Vandenberg's group found that AAV4 and this particular AAV research 32.33, even though this receptor is said to be a universal receptor, some cell types do not need it. And I just go back to idea how we can utilize this idea to enhance gene therapy. And then there are several papers published how we can utilize this knowledge. And one paper published from Korean group published really interesting paper. And they utilize biomaterials coated by this AAVR. And they mix that with AAV and they inject that intramuscularly. And what happens is AAV is like supported by materials. It does not leak into the bloodstream. So that means you can avoid off-target transduction. And also it can make a really high concentration in a particular location in the muscle. So you can enhance transduction. And also other groups co-injected AAVR together with AAV to enhance like a transduction at the neuron synapse and also the transduction in the liver. So that means all of this knowledge can be utilized to enhance gene delivery. And I just would like to switch gears to that another important proteins. So this is the original paper published in Nature that reported AAVR, but in the paper, they also found second hit and unknown hit. This is a GPR108. So this is a G protein coupled receptor protein. And they didn't focus on this one, but more recently, and two groups and focus on this one and clearly show that this is also a multiple cell type AAV entry factors. And they did a really accurate interest study because as I said, and AAV research 30.233 does not require AAVR for transduction. The idea is that they affect this AAVR knockout cell line with this particular cell types. And the idea is if we don't have any important protein knockout, and this virus can transduce the AAVRKO. But if you use a CRISPR-Cas9 library, if you knock out important protein, now you lose the transduction. By taking that approach, they identified this GPR108 as a top hit that is really important for AAV transduction. So at this point, this protein, how this protein and what physiological function of this protein is not really well known. But recently people have reported that this protein may be really important for innate immune response because we are now talking about the virus. The function kind of makes sense. But at this point, and what exact role this protein is playing a role in our body is not known. And I just would like to summarize the two different, how to say, co-playing receptors, GPR108, AAVR. And so many AAVR requires both GPR108 and also AAVR. But as I said, some AAV serotypes does not require AAVR. And interestingly, recently, and Luc Vandenberg's group found that AAV5